Good afternoon, students. Myself, Sucheta Bidve, and our subject is plant biotechnology. Today we are going to complete second part of introduction to biotechnology. Students, today we are going to cover four important topics in which we are going to see importance of plant biotechnology with respect to plant micropropagation, plant mutation cloning, plant cell technology, and environmental biotechnology. Plant micropropagation. As the name indicates, and you are totally familiar with the concept of micropropagation. Okay, micropropagation, it is a simply technique of regeneration of entire plant by using cell, tissue, or organ. It is tissue culture based technique which is rest on principle of totipotency. Now, what is totipotency? Totipotency is a ability of a single plant cell to regenerate into entire plant. Means if we culture a single cell of a plant, it has its own ability to develop into entire plant. Simply. So it is a alternative for a conventional asexual propagation method. Conventionally, the asexual propagation methods are used to produce number of plantlets. And nowadays, we can achieve this by using a most popular technique of plant micropropagation. In plant micropropagation, a small amount of tissue is needed as initial explant for regeneration of millions of clonal plants per year. Means a small amount of tissue is used to prepare millions of clonal plants. It is not depend upon the season. Means generally the sexual propagation methods as well as asexual propagation methods in a plant depends on the season. For example, nowadays it is a rainy season and you might be heard that in rainy season the propagation of plant may be faster. Okay, likewise, in some plants, if the flowering and fruiting is occurring in the summer or winter, then the propagation season will be, yes, summer or winter. But in case of this micropropagation, it is not depend upon season, so it is very quick method of proliferation of plants. Now, when we think about the relation of plant biotechnology and specifically to the plant genetic engineering, there is a very close relation of micropropagation and this genetic engineering, which can be collectively used for the crop improvement in which a variety of economically important plants can be designed for stress resistance, herbicide resistance to increase shelf life of the plant, which leads to increase in productivity of plant and decrease in the loss of production by means of pathogen or environmental stressful conditions. Generally, this can be achieved by means of or by introducing a resistant gene into economically yielding varieties. And after introducing a gene of interest into the plants, there is development of genetically engineered or genetically modified plant. This developed genetically modified plant can be propagated by means of plant micropropagation. When we are thinking about the plant micropropagation, the well-known examples are likewise whenever when we are thinking to develop a stress-resistant plant, the stress in case of plant, the stress may be of two types. It may be biotic stress or it may be a abiotic stress. Generally, biotic stress means the stress caused by the pathogens, and abiotic stress, it is caused in a plant by means of external environmental conditions. Now, we'll take a simple, most well-known example of abiotic stress-resistant plant, BT cotton. Yes, in case of BT cotton, a cryogene was transferred from Bacillus thuringiensis, which is a bacteria, into cotton plant. And due to this construct, the plant is getting resistant against the gallworm, which is a fruit-eating pathogen of the cotton plant, which leads to decrease in economic productivity. But when we use this genetically modified BT cotton, there is a tremendous decrease in disease caused by this gallworm, and which leads to increase in high production of cotton. So this can be achieved by genetic engineering, and so the genetically modified plant which are obtained after genetic engineering can be propagated by means of plant micropropagation. Similarly, we can develop a herbicide. There are examples of herbicide resistant plant also. Now, what is this herbicide resistance? We know what are herbicides. The herbs which are grown in the field, which are useless, should be killed for the good yield of the economically important plant. Okay, initially it was simply done by the physical or manual process of weeding, but nowadays we can achieve this weeding process by simply use of herbicide resistant plant, okay, in which we are going to spray herbicides on the weeds, which leads to destruction of weeds, but it doesn't hamper it doesn't cause any bad effect on our crop. Why? Because our crop were introduced with the herbicide resistant plant. Let's take an example. Tomato, which is resistant to herbicide sulfonyl urea. How it is achieved? The gene expression of specific enzyme which detoxifies the effect of herbicide. Okay, these are the well-known example for the micropropagated for the genetically engineered plant produced via the grown via the micropropagation. Nextly, a most emerging field in case of plant biotechnology is molecular farming. Okay, now in case of molecular farming, the genetically modified plant can be used as a factory for production of specific chemicals or pharmaceutical compounds, in which the plant's genetically engineered plant can be used as a bioreactor. So simply, a desired product is produced within the plant, and it is achieved by means of biotechnology. In order to achieve this, simply this procedure is carried out. A structural gene means a gene of interest which we are going to introduce in which we are interested is initially cloned into plant expression vector, and this plant expression vector is introduced into the plant, which leads to development of transgenic plant. Now, what is use of this transgenic plant, such a transgenic plant? Mainly, these are used to increase the nutritional quality of our food. For example, we'll take an example of potato. We know the potato is rich source of carbohydrates. It doesn't having any amount of proteins. But we can, by means of genetic engineering, we can increase the nutritional value of this potato. Simply, the gene AMAI from the amaranthus species is introduced into the potato, which leads to increase in synthesis of amino acid within the potato. So, it is a well-known example of increase in nutritional quality from the in the potato. Likewise, there is example of golden rice. Golden rice, what is this golden rice? Yes, the golden rice, it is having a precursor for the synthesis of vitamin A. We know vitamin A is essential for the eyes, and this vitamin A synthesis can be triggered by introducing a gene responsible for the synthesis of beta carotene, which is achieved in the golden rice, and it is useful to improve health in the malnourished people in the poor areas. Likewise, we can introduce genes to increase the immunity of the organism or human beings within the edible fruits or vegetables, which leads to introduction of new concept of edible vaccines. In edible vaccines, the antigens are produced into the food material itself, which leads to stimulation of immune system. Okay, means the immunization can be increased, or there is achievement of increase in stimulation of immune system by simply eating a food material. Yes, and it is achieved by means of production of antigens within the edible fruits or vegetable itself. For example, in tomato, there is expression of rabies, rabies glycoproteins, and this rabies glycoprotein are lead to or hope to develop a vaccine against the rabies. Likewise, there is a phenomenon of edible antibodies, edible interferons, etc., which leads to a new revolution in pharmaceutical industries. Okay, have you clear that? So, by using, or when we see a importance of biotechnology with respect to micropropagation, we can say this: by using genetic engineering, we can modify the expression of genes within the plant, and this constructed plants can be propagated by means of micropropagation. Likewise, this micropropagation has a very good application within the forestry. In forestry, if we use plants which are inborn resistant to stress, like a biotic stress and abiotic stress, which leads to development of healthy plants into the forest with very less maintenance. Okay, so it will helpful to increase the land under the forestry. Nextly, the next part is plant cell technology. Now, plant cell technology nowadays, it is an important tool for the crop improvement, commercial production of natural compounds into the plant cell, as well as development of forestry. In plant cell technology, cell or protoplast culture coupled with the DNA vectors to problem caused to overcome problems caused by barriers to gene transfer through sexual propagation means. Okay, means in case we know that for in order to get a new hybrid plant, there is utilization of methods of sexual reproduction. Now, within this method, it may happen there are barrier genes which are not allowing the process of sexual reproduction. So, this barrier can be removed by means of plant cell technology by fusing or by plant protoplast technology, which is alternatively used for crop improvement. Plant cell culture mainly useful for the production of secondary metabolites from the plants. Generally, the plant cells producing secondary metabolites are isolated and cultured to get enhanced production of secondary metabolites. And nowadays, in the period of corona, we are very familiar one of the drug called as named as hydroxychloroquine. This hydroxychloroquine is mainly a quinine which is isolated or which is synthesized within the Cinchona species naturally. We culture these cells of Cinchona species for production of alkaloid quinine, and we know this quinine is a very good anti-malarial drug, and nowadays it is also effective against the coronavirus. Likewise, there is production of Taxol from the Taxus species. So, for the production of secondary metabolites, we can use a technology for the culture of plant cells. Likewise, simply we can use this plant cell technology to increase the efficiency of biological nitrogen fixation. This can be achieved by introduction of NIF and node genes into the nitrogen fixing plants, into the non-nitrogen fixing plants. We know this NIF gene and node gene, it is important for the biological nitrogen fixation. Mainly these are present into the plant who are having presence of root nodules. Okay, these genes were isolated from this root nodule plants and then it is transferred within the non-nitrogen fixing plant, which leads to increase in ability of biological nitrogen fixation within the non-nitrogen fixing plants, which gives nourishments to plant alternatively. Nextly, application or importance of plant biotechnology with respect to plant mutational cloning. Now, first of all, what is mutation? Yes, mutation is a sudden heritable change in characteristic of the organism. There is mutations may be caused by natural or it can be introduced within the plant. On the basis of that, mutation are of two types. One is spontaneous mutation, other are induced mutations. Spontaneous mutations are natural and they are caused at very low frequency. On the other hand, the induced mutations are artificial and this can be utilized for the crop improvement. A sudden changes are introduced within the plant artificially, which is in which leads to some mutation, some changes into the plant. The required favorable changes can be used for the crop improvement. Likewise, this mutation cloning can be used to study gene expression. Specific function of the gene can be studied by using mutation cloning. Now, in case of mutation cloning, the specific gene's expression is altered, and then the change in a function is studied by means of mutation cloning. Likewise, in case of biosynthetic pathways of secondary metabolites, the gene function can be understood by mutating that specific gene, and this is utilized to construct the pathways or pathways, which is collectively called as metabolite engineering. Okay, so this plant mutational cloning can be effectively used for the crop improvement in order to study the gene expression and for constructing a pathway of secondary metabolite. Next, nowadays we are facing a very severe problem of pollution. We know there are three main types of pollution: air, water, soil. The causes of these pollutions are overpopulation, industrialization, increase in number of vehicles, urbanization, where leads to increase in pollution level of air, water and soil. So here the environmental biotechnology employs the application of genetic engineering to improve efficiency of microbes or other living system to reduce environmental burden of toxic substances. This can be collectively referred as a bioremediation, in which we are using microorganisms or any other living system like algae, fungi, plants, bacteria to clear environmental pollutants. This is nothing but the environmental biotechnology. This environmental biotechnology mainly referred or mainly divided as phytoremediation or use of genetically modified microbes to degrade the pollutant. The phytoremediation is mainly based on the inherent ability of plants to absorb or to clean up the pollutant. This inherent ability of plants mainly based on the root system of the plant, in which there are various other methods or various types like phytoextraction, phytoaccumulation, then the phytovolatilization, etc., in which the plants are going to absorb pollutant from the water, from the soil, then they are going to deposit or accumulate this pollutant within the plant body itself. So it is a phytoremediation. While we can genetically modify the microorganism to degrade the environmental pollutant, and such as genetically modified microbes can be used to clear the industrial waste, like the wastewaters containing hydrocarbons, heavy metals, dyes, xenobiotic components can be separated by using such a genetically modified microorganism. So here the role of biotechnology is development of genetically modified microbes in order to degrade the pollutant, or it is using an inherent ability of phytoremediation. Okay, so these are some importance of biotechnology with respect to plant micropropagation, plant mutational cloning, then plant cell technology and environmental biotechnology. So overall, students, in first part and second part, we have studied the importance of biotechnology with respect to environment, genetic engineering, then micropropagation, mutational cloning, and the cell technology. So, in next lecture, we'll move forward for our next part of the biotechnology. Here are some references which I was used for the preparation of this lecture. You can easily find these references in our library or online. So, if you have any doubt, please ask me. You can find me on my email, or you can ask your questions in our Google Classroom also.